The Constitution of India, Preamble. We, the people of India, having solemnly resolved to constitute India into a sovereign, socialist, secular, democratic republic and to secure to all its citizens justice, social, economic and political, liberty of thought, expression, belief, faith and worship, equality of status and of opportunity, and to promote among them all fraternity, assuring the dignity of the individual and the unity and integrity of the nation. In our Constituent Assembly, this 26th day of November 1949, do hereby adopt, enact and give to ourselves this Constitution.
Hello children, how do you do? Hope you are healthy and happy. Before beginning the session, I want to tell something about gratitude. Gratitude is not a feeling, it is a state of mind that has to be developed, as it taps us into the reservoir of unlimited positive energy. Better sleep, the ability to express kindness, feeling of being more alive, as well as strong immune system are the benefits of showing gratitude. So my dear children, you have to be grateful for each and every moment of your life, as God has given us such a beautiful life. By saying this, I move on to the session where we are going to recap the previous class. So in the previous class, we understood about tissue system. A group of tissues performing a similar function, it is called tissue system, and based on the structure and location, there are three types of tissue system we can see in the plants. Number one is epidermal tissue system, number two ground or fundamental tissue system, and number three vascular or conducting tissue system. First one, epidermal tissue system, which we already understood in the previous class. It is composed of epidermis, stomata and epidermal appendages. The first one is epidermis. In the previous class we came to know about epidermis. It is outermost layer. It is formed of elongated living parenchyma cells, and the epidermis of root is called epiblema. The outermost part of the epidermis is covered with a waxy layer called cuticle, and cuticle is absent in root. Number two is stomata. Stomata, they are the minute pores present in the leaf, and each stoma is guarded by two bean shaped cells called guard cells. And then we understood that guard cells regulate the opening and closing of stomata. You can see the figure related to stomata. In the case of dicot, it is bean shaped, and in the case of monocot, it is dumbbell shaped. And next thing related to the epidermal cell, we came to know that it is associated with guard cell, and that cells are called subsidiary cells or accessory cells. And the function of stomata is, it helps in transpiration, it helps in gaseous exchange. I think, my dear children, the previous class I have taken this part, and I think you would have understood about it. And now the next part is related to epidermal appendages. They are of two types, that also we understood in the previous class, and they are root hair and trichomes. Root hair is concerned with root and trichomes are concerned with stem. The second one is related to ground tissue system, where we understood that tissues except epidermis and vascular bundles constitute the ground tissue, and it is consists of parenchyma, sclerenchyma and collenchyma cells, isn't it? In the case of leaves, ground tissue is composed of thin wall chlorenchyma cells called mesophyll. Third one is vascular tissue system. It consists of vascular bundles, and vascular bundle is formed of xylem and phloem, that also we understood, and in the previous class we came to know about the classification of vascular bundle. and based on the classification we understood that radial, conjoint, radial, conjoint and concentric are the vascular bundles. In the case of conjoint, again we have seen collateral and bicollateral. In the case of collateral, again we have seen open type of vascular bundle and closed type of vascular bundle. In the case of concentric, we have seen again two type of vascular bundles, and based on the vascular bundles we have seen the figures also. It is very important, you have to study, my dear children. Next one, we understood the anatomy of dicot and monocot plant. In the case of dicot root, the thin transverse section, when it is been taken, the layers which is seen in the dicot root are epidermis, cortex, endodermis and stele. Epidermis, it is outermost layer. Cortex, it consists of thin walled parenchyma cells. You can see the figure, and that also we studied in the previous class, that is related to cortex. Next we understood about endodermis, it is the innermost layer of cortex, and here the wall is thickened due to the deposition of suberin, and because of that it is impermeable to water, and this thickening is called Casparian strip or Casparian thickening. It is very important. And in the endodermis we can see passage cells, and these passage cells are helping to allow the passage of water from cortical cells to xylem. And next one is stele or stelar region. It is the central portion of root, and it consists of pericycle, vascular bundle, pith and conjunctive tissue. That also we understood in the previous class. In the case of pericycle, it is inner to the endodermis. Next one is vascular bundle, which is very important. They are radial and bundles are seen in 2 to 4 in numbers. Xylem is exarch, that also we understood. Then next one is the conjunctive tissue, which is found between xylem and phloem. The last part is pith. It is very small. You can see this figure, and I think you would have understood by seeing the figure. Next one is related to monocot root, which already understood in the previous class. Here also a transverse section when taken, we have seen epidermis, cortex, endodermis, stele are the regions. Epidermis, again it is outermost layer and it is single cell structure. Cortex consists of parenchyma cells. Same way endodermis, Casparian strip and passage cells are present. In the case of stele, pericycle, vascular bundle, conjunctive tissue and pith is present. In the case of vascular bundle, it is radial, xylem is exarch, it is polyarch condition, that is very important. And I think by seeing the figure you would have understood about this part also. And the third, we have seen the anatomy of dicot stem. Again by seeing the figure we have understood that it is also having epidermis, cortex, hypodermis, general cortex, endodermis, isn't it, as well as the stelar region as well as the pith. In the case of epidermis, it is outermost layer. In the case of cortex, it is being divided into 3 regions: hypodermis, general cortex as well as endodermis. And then we understood that in the endodermis there are starch grain, therefore this layer is also known as starch sheath. And next is stele. In this pericycle we understood, and later on we understood about bundle sheath, that is vascular bundles, and here we have understood that it is composed of xylem, phloem and cambium, and the vascular bundle is limited and they are arranged in ring. Later on we understood that they are conjoined, collateral, open and endark. And next one we came to know that medullary rays are also present there, and the last is the pith, which is very large and it is central. So that was related to the previous class. Today we are going to study and understand about anatomy of monocot stem. You can see the figure of monocot stem and you can go through your textbook. This is very important. This is the ground plan, this is the vascular bundles, and here you can see the TS or enlarged portion. Here it is said that the following distinct parts are shown here: epidermis, hypodermis, ground tissue and vascular bundle. In the case of epidermis, already we know that it is outermost layer and it has numerous stomata which helps in the exchange of gases. Hypodermis, again it is below to epidermis and it helps to give mechanical support, and in the case of ground tissue it consists of soft parenchyma tissues and it is concerned with the storage of food material. In the case of vascular bundles, which is very important, here we can see each vascular bundle is oval, conjoint, collateral and closed, showing endark xylem. This is very important. And medulla is the pith, medullary rays and pericycle, they are absent in the case of monocot stem. So, that is related to monocot stem, my dear children. Next, we are going to study about the anatomical difference between dicot stem and monocot stem. You can see in the case of hypodermis, in dicot stem it is made up of collenchyma, in the monocot stem it is made up of sclerenchyma cells. In the case of ground tissue, it is differentiated into cortex, endodermis and pericycle as well as pith. In the case of monocot stem, not differentiated, but it is a continuous mass of parenchyma. In the case of starch sheet, it is present in dicot stem, it is absent in monocot stem. And in the case of medullary rays, it is present in dicot stem, it is absent in monocot stem. Now, related to vascular bundle, which is very important, related to dicot stem as well as monocot stem. In the case of dicot stem, it is said that it is collateral and open, arranged in a ring, and secondary growth occurs in dicot stem, whereas in the case of monocot stem, it is also collateral, but it is closed, it is scattered in the ground tissue and secondary growth does not occur. So, that is the difference between dicot stem and monocot stem. This is very important, my dear children, you have to study it. Next one is related to the anatomy of dicot leaf. So, till now we have understood that there is an anatomy based on stem as well as root. Now, we are going to study about the internal structure of dicot leaf. In the cross section of dicot leaf, we can see the distinct region. Here also we can see epidermis and next is mesophyll layer, and in the case of epidermis, upper and lower epidermis, they are covered with cuticle and both the epidermis bear stomata, and the next layer is the mesophyll layer. This tissue, or we can say the tissue seen between upper and lower epidermis is called mesophyll, and it is differentiated into palisade parenchyma and spongy parenchyma. You can see the figure here, this is the anatomy of a dicot leaf. In the case of palisade parenchyma, they are seen on the, this is upper epidermis, which is also called adaxial epidermis, and this one you can see this is the palisade parenchyma, and they are elongated cells, they are arranged parallel to each other. These are the elongated cells, they are arranged parallel to each other. Next one is the spongy parenchyma. You can see these are the spongy parenchyma cells, and they are oval and they are loosely arranged. They are loosely arranged, that means we can see the intercellular space between the spongy parenchyma cells, and they lie towards the lower epidermis. This is the lower epidermis, and this is also called abaxial epidermis. So, that is related to palisade and spongy parenchyma. And next one, we can see the vascular bundle present in the dicot leaf. These are surrounded by bundle sheath cells, and the vascular bundles are collateral and closed. Xylem is seen on the upper side and phloem is seen on the lower side. You can see this is phloem, okay, this bluish color is phloem, and this one is the xylem, where you can see this one is the protoxylem and this one is the metaxylem. So, metaxylem is facing towards the pith and protoxylem is facing towards the periphery. Okay, so you can see here protoxylem facing towards the upper epidermis. This is upper epidermis, so it is facing towards the upper epidermis. I think you would have understood about it. So, that is related to the dicot leaf. Now, we are going to understand and learn about the monocot leaf. Here also we can see three distinct regions. Epidermis. In the case of epidermis, again it has been said that upper and lower epidermis are present there and they are coated with cuticle. Stomata are present on both the epidermis. The upper epidermis contains special bulliform cells. This is very, very important. Bulliform cells. Bulliform cells are also known as motor cells. They are large, empty, colorless cells found in the adaxal epidermis in grasses. Adaxal means in the upper epidermis in the grasses we can see large, empty, colorless cells, and these cells are called bulliform cells or motor cells. And when these bulliform cells they absorb water, they become turgid. When these cells they absorb water, they become turgid. As a result, the leaf surface is exposed. Now, due to water stress, when water is not there, surely these cells they become flaccid. You can compare it with a balloon. When we fill air in the balloon, it will become turgid. When the air is taken out from the balloon, it becomes flaccid. The same thing we can see in the case of bulliform cells. So, when the water stress is taking place, bulliform cells become flaccid, and as a result they make the leaf curl inward so that the water loss will be minimized through the leaf. That is the main function of bulliform cells present in the leaf. Next one is the mesophyll layer. It is not differentiated into palisade and spongy layer. You can see here no difference between any palisade layer or spongy layer. Next one is vascular bundle. They are collateral, closed and endark. Each bundle is covered by bundle sheath. They are closed, that is no cambium is present there. They are collateral as well as the xylem is endark. I think you would have understood about it, my dear children. Now next one is the difference between dicot and monocot leaf. In the case of nature of orientation, dicot leaf is typically dorsiventral. In the case of monocot leaf, it is typically isobilateral, isn't it? Two sides we can see same. Next one is stomata. In the case of stomata, it is hypostomatic, and in the case of monocot leaf, it is amphistomatic. So, more stomata we can see on the upper side of the leaf, and in the case of monocot leaf, on both the side we can see stomata. Next one is motor cells. They are absent in dicot leaf as they are present in monocot leaf in the upper epidermis. In the case of mesophyll, it is differentiated into palisade as well as spongy parenchyma. In the case of monocot leaf, it is undifferentiated. In the case of the presence of vein, irregularly scattered and they are parallel arranged. In the case of xylem vessel, in dicot leaf, protoxylem and metaxylem vessels in each bundle are many. In the case of monocot, 2 protoxylem and 2 metaxylem bundles are seen in each bundles. Protoxylem and metaxylem vessels are seen in each bundles. Okay, so that is related to the difference between dicot and monocot leaf. That is again important, you have to study it. Now we are going to study and understand the next topic, that is secondary growth, which is also known as secondary thickening. What is primary growth? It is increase in the length by growth. When the plant is growing in length, we can say primary growth. It takes place by the activity of apical meristem. Already we know that what is apical meristem. Now next one, what is secondary growth? It is increase in the girth, or we can say thickening by growth, that is called secondary growth, and it takes place by the activity of lateral meristem. We know that what is lateral meristem, isn't it? That is vascular cambium and cork cambium. The secondary growth due to the activity of vascular cambium is called stellar secondary growth, and if this growth is due to the cork cambium, it is said to be extrastellar secondary growth. So, when the secondary growth is taking place, it is taking place because of the activity of vascular cambium as well as cork cambium. If vascular cambium is responsible for the secondary growth, we can say it as stellar secondary growth, and if the cork cambium is responsible for the secondary growth. We can say it has extrastellar secondary growth. I think you would have understood about stellar and extrastellar. Now we can see how secondary growth is taking place in dicot stem. It includes stellar secondary growth, extrastellar secondary growth. Stellar secondary growth, in the stellar secondary growth we can see it takes place by the activity of vascular cambium. And what is vascular cambium? The meristematic layer that is responsible for cutting of vascular tissues, xylem and phloem. There is a meristematic tissue or there is an active zone which is cutting of vascular tissues, that is xylem and phloem, that is called vascular cambium. So, that meristematic zone or the active zone which is cutting of the vascular tissues as xylem and phloem, that zone is or that is called, that layer is called vascular cambium. Now, next one you can see secondary growth in dicot stem includes the following two steps. Taking into two steps. Number one, how the secondary growth we can see in dicot stem. Number one, formation of cambial ring. Number two, activity of cambial ring. First one is formation of cambial ring. You can see the figure here, my dear children. The cambium present between the primary and the second primary phloem and primary xylem is called intravesicular cambium. You can see here these are the vascular bundles and in between you can see these are the intravesicular cambium. Okay, intravesicular cambium, which is also known as vesicular cambium. These are called intravesicular cambium or they are also called vesicular cambium. Now, next one, the cells of medullary rays adjacent with the vesicular cambium becomes meristematic to form a new cambium and it is called intervesicular cambium. Now, what happens between these vascular bundles, between these two vascular bundle, these two vascular bundle, these two vascular bundle, there are some meristematic cells or we can say medullary rays become meristematic. And due to that what happened, new cambium is formed there. You can see in this figure between these two you can see new cambium is formed there. These two new cambium, to these two bundles new cambium. Again you can see here, so complete a cambium ring we can see there. So this type of new formation of new cambium is called intervesicular cambium. So between the bundles if cambium is formed there, that is called intervesicular, and in the bundle, in a single bundle if the cambium is formed there, that is called vesicular or intravesicular cambium. So in this manner formation of cambial ring is taking place, my dear children. That is the first step in the secondary growth which is taking place in dicot stem. Now what happens later on, both these cambium unite to form a complete cambium called cambial ring, isn't it? You can see here. This is how cambial ring is formed there. Now the next one is activity of cambial ring. In the case of activity of cambial ring, you can see the ring of vascular cambium cuts off cells both on outer as well as inner side, and the cells which is cutting on the outer side becomes the secondary phloem and the cells which are cutting to the inner side becomes a secondary xylem. Here you can see this is secondary xylem is formed here, secondary phloem is formed here, isn't it? This is secondary phloem, this is secondary xylem and this is the cambial ring. Okay. Now the amount of secondary xylem which is cut off there is more than the secondary phloem. Thus what happens, there takes place the formation of secondary tissue and due to that increase in the girth or the diameter occurs and this is called secondary growth. As more secondary xylem is cut off, so what happened, there will be increase in the girth taking place and that is called secondary growth. And the cambium cells, they are rectangular, thin walled and they are having meristematic activities. So that is related to the activity of cambial ring. What a cambial ring is doing? It is cutting off the cells again to the outer side as well as to the inner side, and the outer side of the cell which cells are formed, they are the secondary phloem, and to the inner side the cells which are formed, they are secondary xylem. As secondary xylem is formed more in number as compared to the secondary phloem, surely there will be increase in the diameter, or we can say increase in the thickness or increase in the grit taking place, and this is called secondary growth which is taking place in the stem. Now next, related to the next topic, that is spring wood and autumn wood. Based on secondary growth taking place in the stem, we can see two types of wood, spring wood and autumn wood. You can see here spring wood is also called early wood and autumn wood is also called late wood. In the case of spring wood, it is formed during spring season. Late wood, it is formed during winter season. It is formed early in the year, it is formed after the early wood, and it consists of xylem tissues with wider vessels, and the late wood or autumn wood consists of xylem elements with the narrow vessels. This spring wood is produced more in amount and late wood is produced less in amount. They are less dense and late wood are more dense. Spring wood, they are having broad zone of wood, whereas late wood are having narrow zone of wood, and the spring wood, they are not as strong as late wood, whereas late wood, they are stronger than early wood due to the large volume of wall material. So this is the difference between spring wood and autumn wood. You can see the figure related to spring wood as well as autumn wood. You can see here dark narrow circle which is seen there, dark narrow circle which can be seen there, they are the autumn wood, and spring wood, they are the light wider circle. The lighter wider circle which is showing there, that is spring wood. That is the difference between early wood and late wood, or we can say spring wood or autumn wood. My dear children, you have to study it, it is very, very important. Now as the spring wood and autumn wood is formed there due to the formation of or due to the secondary thickening taking place in the stem, because of that we can see annual rings present in the stem. What is annual rings? Xylem formed by the vascular cambium during one growth season, that is annual ring. Xylem which is formed by the vascular cambium during one growth season is the annual ring, and annual ring, it constitute early spring wood as well as late summer wood. What is early spring wood? Vessel diameter is large, xylem walls are thinner. And what is late summer wood? Vessel diameter is, that is xylem vessel diameter is small and the wall are thicker. In the case of early spring wood, xylem vessel, its diameter will be large. In the late summer wood, the xylem vessel diameter will be small. And next thing, it is been said that tropical trees have no annual rings because seasons are so similar. In the case of tropical trees, seasons are similar, therefore they do not have any annual ring seen in the wood. So that is about annual ring. Now next one is, we can see heartwood as well as sapwood also in the stem. Sapwood, it is formed outer, it is, or we can say it forms outer wood of an old stem. Heartwood, which is also called duramen, it forms the central wood of an old stem. When we are taking the old stem and we are cutting it, we can see the central part is being covered by heartwood, and the peripheral part or the outer wood which is shown there, which is lighter in color, is the sapwood. Sapwood is light in color, heartwood is dark in color. Sapwood is having lighter, they are lighter in weight, heartwood is heavier in weight. Sapwood, it contains living cells, heartwood, living cells are absent. It is a functional, sapwood is a functional part, whereas heartwood is dead and non-functional part. In the case of sapwood, tyloses are absent. In the case of heartwood, tyloses are present. So these are, tyloses is a material which makes the wood more stronger. So in the case of Heartwood tyloses is present and makes the wood more stronger, which is present there in the heartwood, and this tyloses is absent in the sapwood. So that is the difference between heartwood and sapwood. That is also important, you have to go through it. Next one is extrastellar secondary growth. Till now we have studied about stellar secondary growth, and we came to know about the through stellar secondary growth, the secondary growth is taking place in the stem, and based on that thing we have seen heartwood and sapwood, we have seen springwood and autumnwood, annual ring, all these are connected with each other with a secondary thickening in the dicot stem, isn't it? Now second one is extrastellar secondary growth. This takes place by the activity of cork cambium. And what is cork cambium? It is also called phellogen. It is a secondary lateral meristem which originates in the extrastellar region. And what is extrastellar region? It is the cortex. So in the cortex, secondary lateral meristem is originating, and that is called cork cambium, or we can say phellogen. It produces two types of tissues. One is the cork, which is called phellum, and the other one is the secondary cortex, that is called phelloderm. The first one is cork. It is the dead tissue formed outside the cork cambium. Outside the cortex, we can say if dead tissue is formed there, that is called cork, or it is also called phellum. And next one is secondary cortex or phelloderm. It is the living parenchyma cell, and it is formed inner to the cork cambium. You can see this is the structure of the stem, where we can see this is epidermis. From here till here it is phellum. Okay, that is cork, and this part is phellogen. This part is phellogen. And from here till here it is secondary cortex, or we can say phelloderm. So phelloderm is living parenchyma, and it is formed inner to the cork cambium, and we came to know that cork is a dead tissue which is formed outside of the cork cambium. That is related to the phellum as well as phelloderm. Now phellogen, phellum and phelloderm, they are collectively called periderm. That is again very important. Phellogen, phellum, isn't it? What is phellogen? Phellogen is cork cambium. What is phellum? It is cork. And what is secondary cortex, or we can say phelloderm? It is secondary cortex. These three constitute the periderm. So that is related to periderm, which is very important, my dear children. You have to study it. This figure is also important. Now next one is related to bark. It refers to all tissues exterior to vascular cambium. All the tissues which are coming there other than or except vascular cambium is referred to as bark, and it includes secondary phloem and periderm. Periderm already we came to know what is periderm. With that, secondary phloem is also coming there. So secondary phloem and periderm includes bark. Bark formed early in season is called early or soft bark, and the bark which is formed at the end of the season is called late or hard bark. So there are two types of bark, soft bark and hard bark, or we can say early bark or late bark. When the bark is formed early in the season, it is called early bark. When the bark is formed at the end of the season, it is called late bark. You can see here, this is bark. Okay, my dear children. Now the next one is lenticels. These are lens shaped opening formed in the cork. We have seen in the figure related to lenticels. These are in the bark, we can see like this kind of structure. They are lens shaped, and you can see here one portion is shown here. This part is a lens shaped. When we are going near to a big tree, it is having a bark, and in the bark if we are able to see lens shaped structure, they are called lenticels, and they help in exchange of gases. Lenticels help in exchange of gases. In the case of leaf, stomata, they helps in exchange of gases. In the case of stem, we can say lenticels are helping in the exchange of gases. So, this we can say it has in the form of base pair relationship. Stomata is to leaf, dash is to stem. In the case of stem, it is lenticels. In the case of leaf, it is stomata. I think you would have understood about lenticels. Now, the last part of the chapter, that is secondary growth in root. It takes place by formation of Campbell ring, and number 2 by the activity of Campbell ring. In the case of root, we can see the Campbell ring, how it is formed. You can see here, it is wavy in the beginning, unlike in dicot stem. In the dicot stem, we have not seen wavy structure, isn't it? And the Campbell ring is completely secondary in origin, as cambium is absent in monocot. So, in the secondary thickening, when the secondary thickening is taking place, that time only the Campbell ring is taking place. So, here you can see wavy structure in the beginning, isn't it? And later on, it is becoming the circular structure. So, here it has been said the conjunctive tissues inner to phloem becomes meristematic to form cambium. In this case, what is happening? Inner to the phloem, conjunctive tissues are present there, which is becoming meristematic, or we can say which is becoming very active, and cell division is taking place there. And the pericycle cells outside the protoxylem becomes meristematic to form the stripes of cambium. In the case of formation of stripes of cambium, pericycle cells, they are becoming meristematic. The pericycle cells, they becomes meristematic, and due to which the stripe of cambium is taking place, and thus Campbell ring is formed there. These Campbell strips unite to form a wavy Campbell ring. We can see here, this is epidermis, endodermis, this is pericycle, this is primary phloem, this one is the cambium. Already I told you about it, that is inner to the phloem becomes the conjunctive tissues, inner to the phloem becomes a meristematic, and cambium is formed there. So, that is related to this part, isn't it? And this is primary xylem. Now, the second part you can see here, a wavy ring is formed there, isn't it? This wavy ring is Campbell ring. Now, next one, activity of Campbell ring. Once the Campbell ring is formed there, we can see what is the activity taking place by the Campbell ring. The cells of Campbell ring cuts off towards outside and inside. The Campbell ring which is formed there, it starts to cut outside as well as inside, and the cells formed outside becomes secondary phloem, and the cells which is formed inside becomes the secondary xylem. As the same activity we have seen in the case of secondary thickening taking place in dicot stem, the same activity we can see related to the Campbell ring in the case of the secondary thickening taking place in the roots. So, here again you can see it is been said that the Campbell ring will be cutting off towards the outer side as well as towards the inner side. The cells which are formed outside is forming the secondary phloem, and the cells which are formed inside is forming the secondary xylem, and in this manner secondary thickening or secondary growth is taking place in the case of roots also. So, I think this is the structure related to the secondary thickening taking place in the case of root. You can see here epidermis, cortex, endodermis, and here you can see primary phloem, this is secondary phloem, isn't it? This one is the Campbell ring. Again you can see this is primary xylem, and this is primary xylem, and this one is the secondary xylem. So, like this formation of the complete secondary thickening is taking place here because of the activity of Campbell ring in the root. I think, my dear children, you have understood about the secondary growth taking place in root. My dear children, this was the last topic of chapter 4. I think you would have understood chapter 4 clearly. You have to go through today's topic 2 to 3 times, then only you will be able to understand it very clearly. If you are having any doubt related to chapter 4, you can call me and clarify it. Please be on track and always show gratitude towards others. Okay, my dear children, by saying this, I wind up this session and we will be meeting in the next session. Till then, take care.